बच्चों, let's see the question. So the question says the restriction enzyme echo R1 has the property of. So इन चारों ऑप्शन में से हमें echo R1 के property को identify करना है. So the key concept in this question is to understand what is echo R1. So this is a fairly easy question, and echo R1 is actually a restriction endonuclease. So it's an enzyme that can cleave DNA at a specific site, and therefore it is known as the restriction endonuclease. So this enzyme is isolated or derived from E. coli. So this restriction enzyme is found in the bacteria E. coli and the RD strain of the E. coli. And therefore the name EcoR1 comes from the origin of this restriction endonuclease. So now if we see how the restriction endonuclease cleaves the DNA, then so firstly the recognition site which is recognized by restriction endonuclease is GAATTC, and its complementary strand as CTTAAG. So this is the recognition site which is recognized by the enzyme EcoR1. So now this EcoR1, after recognizing this recognition site, cleaves the DNA into two fragments at this point and this point. So therefore, we will get two fragments of DNA as follows. So this will be the two fragments of DNA formed due to the cleavage by the restriction endonuclease, that is EcoR1. Now, the EcoR1 results in the formation of sticky ends. So, the overhanging flanks of the nucleotide sequence present in the two new DNA fragments which are formed due to the cleavage by the EcoR1 is known as the sticky ends. So this one is the sticky end and this is the other sticky end. So these sticky ends can now be complementary to the other sequences and then can bind with other DNA fragments with the help of DNA ligase. So this is how EcoR1, the restriction endonuclease, function. So EcoR1, we can say that it has an endonuclease activity. So now if we see the options given, so the first option says endonuclease activity. So we saw that EcoR1 has the property of endonuclease activity. So this is the correct statement. Then the second statement says exonuclease activity. So this is incorrect because EcoR1 cleaves the DNA in the middle region and not at the ends of the DNA as observed now. Now the third option says ligation activity. So ligation activity is also wrong because DNA ligase is responsible for that. and not eco-R1. And the fourth option says it is correcting the topology of replicating DNA. Again, this is also incorrect because that's the exonuclease activity and not an endonuclease activity. So our final answer for this question is option number 1, that is echo R1 has the endonuclease activity. So this is our final answer. Hope you understood this well. Best of luck. Thank you.